Hello everyone, my name is Himani Agarwal and today the topic which I am going to discuss is transcription in prokaryotes, and it is going to be a detailed explanation, and it's beneficial for all the BSc and MSc students and also the students who are going to appear for NET or GATE or any other competitive examination. So let's begin. These are the topics which I am going to discuss with you guys. So let's start them one by one. What is transcription? Transcription in basic English term is a word which is used to define to copy, to transcribe something to something else. In science, transcription is the process by which DNA is synthesized into RNA. Alright? And this is a very important process, and it is an intermediate process by which DNA can be converted into proteins, which are the actual functional components of a living being. In prokaryotes, this process occurs in the cytoplasm. Please remember this, it is a very important point asked many times. It occurs in the cytoplasm alongside translation, that is, as soon as DNA is converted into single-stranded RNA, it is then synthesized into polypeptide chains or also called as proteins. Okay, which are called as proteins, which are the functional components of a living organism. So, what is the need of this process? Why not DNA directly be converted into protein? This thing should come into your mind. Why? Because DNA cannot just leave its origin and go to different parts in the cell and then function as protein. That's why it requires an intermediate in the form of RNA, which can be converted into protein. Okay, so this is the first step in which the genotype is converted into phenotype in a living organism. The major components of this process are DNA template, the substrate for transcription, and the transcription apparatus. Let's discuss them one by one. DNA template. See, before coming to this, we should know, during replication, both the strands serve as template, right? Both the strands are replicated into daughter DNA strands. But in case of transcription, this is not the case. In case of transcription, out of the two strands of DNA, only one strand serves as the template, which is synthesized into RNA. Okay. That strand which is converted into RNA is called as the template strand, and the other strand which is not transcribed is called as the non-template strand. The template strand is in the direction of 3' to 5', and the non-template is 5' to 3'. In the template strand, we need to see the transcription unit. The transcription unit is the unit which actually gives rise to our RNA molecule. Let's remove this ink. In transcription unit, we should know the three regions. First is the promoter region, second the RNA coding region, and the third is the termination site. Promoter region. What is a promoter? Promoter is something which promotes something, which initializes something. So, promoter is the region where the transcription apparatus comes and binds to. It is the first region which comes into the eye of transcription apparatus. From here, when the transcription apparatus crosses this region and enters into the transcription site. This is called as the RNA coding region. This is the region actually which gives rise to our RNA. Third is our termination site. Termination site is the end of the row. After this, the transcription apparatus will not proceed further and go to the next DNA sequence. No, it will not. But we should remember this termination site is also transcribed into the RNA. You can see this red region? Yes. So from the transcription start site, which has been indicated as +1, the first nucleotide which is converted into RNA to termination start site is our RNA molecule. Okay, this is our transcription unit. Coming next to substrates for transcription. Okay, suppose we are cooking something. Suppose we are making tea for someone. We need milk, we need sugar, we need the basic essential requirements to make tea. Similarly, to make something new from a DNA strand, obviously we are making something new. From DNA we are making RNA, right? We need something, right? We need some substrates, we need some raw materials. These raw materials are ribonucleotide triphosphates. In short form, it is written as RNTPs. RNTP and DNTP. RNTP is used in RNA. DNTP is used in DNA synthesis. Alright, what can we see the basic difference between RNTP and DNTP? See, only the hydroxyl group which is present at 2' region. Instead of hydroxyl here, we will see one hydrogen molecule. That's why it is called as deoxynucleotide triphosphate. A lack of one oxygen molecule. Okay? So, ribonucleotide triphosphates. These are the triphosphates which participate in RNA synthesis. This is alpha, beta, and this is gamma phosphate. Alright? Out of these three phosphates, only the alpha phosphate participates in phosphodiester bond formation. The other two are released. So in a growing polypeptide chain, which is from 5' to 3' region, we have seen the others on the past slide. 3' means there is one hydroxyl molecule, right? So this hydroxyl molecule will participate with this alpha phosphate, and this will lead to phosphodiester bond formation. Okay? Phosphodiester bond formation. And then this RNTP gets attached here. Okay? So next again one more RNTP will come, and in the similar way it will get attached to the next hydroxyl molecule. This is how it happens. But here we can see in the 5' region, of a growing chain, this is an important point, okay? You should remember this. 3 molecules of phosphate are not removed. Here you will see, here in this region you will see 3 phosphates itself. 3 phosphates. Why? Tell me why? Because they are not participating in any kind of bond formation, right? Hydroxyl group of this first RNTP will go and attach to the second RNTP. The first RNTP of this growing chain is a triphosphate. Only the 5' region is triphosphate in nature. Now, coming to the apparatus of transcription. The main Apparatus are the enzymes which participate in any biological function. Similarly, as DNA replication, we have many enzymes, right? In transcription for prokaryotes, there is only one enzyme which is very important. But in case of eukaryotes, the case is different. In case of eukaryotes, there are three different RNA polymerases which have different functions. But let's not go there. Today we'll just be seeing RNA polymerase of prokaryotes. In case of prokaryotes, it is a 5 subunit structure. Alpha, which are 2 in number, beta 1, beta prime 1, and a sigma factor, which is also 1. But these four together are called as core enzyme. Together with the sigma factor, it is called as the hollow enzyme. Okay? Together with the sigma factor, it is called as the hollow enzyme. These subunits, when a gene is being transcribed, will come together, assemble. When their function is done, they will disintegrate. Again, when a gene is required to be transcribed, again they will come and rearrange themselves together. When their job is done, again they will disintegrate. This is how they function. Each subunit of this RNA polymerase has a different role to play. For example, this alpha subunit has a role in promoter recognition, similar to a role to sigma factor. Okay, beta and has the role in DNA elongation, and beta prime has a role in DNA binding. Beta prime enhances the binding of RNA polymerase to the DNA so that it can be transcribed. It enhances the binding of RNA polymerase to DNA to be transcribed. So next we will be discussing about the process as a whole. This process will be seen in second part of transcription in prokaryotes. Thank you.